and good morning to you all. I'm going to take you in the next few minutes through what we're doing at eTheRNA to broaden the applicability of mRNA-based therapeutics. There's of course no question that the field has exploded recently with the phenomenal success of the SARS-CoV-2 vaccines and is moving into therapeutic areas previously occupied by other modalities in biopharmaceuticals. And of course, these are in some cases quite early stages and are not probably benefiting also from the the accelerated approval process that SARS-CoV-2 vaccines have benefited from. But nevertheless, there is a lot of activity, and this is obviously reflected also in value predictions on how the market size is going to increase over the next few years. Now, on this slide, this is by no means exhaustive. I have listed some of these new areas, and next to it, I put perhaps some of the challenges that remain in moving into some of these areas. And I've highlighted in blue where perhaps these challenges are down to technology or delivery technology, and where these limitations are, could be overcome by engineering or improving these technology platforms. So this is to do with targeted delivery, for example, also the avoidance of any inflammatory activation, the tolerability, of course, as well, and in some cases, the longevity of the effects. And also, of course, as we know from the SARS-CoV-2 vaccines, the stability and also the cold chain challenges are also some aspects that affect certain therapeutic areas. So at eTheRNA, our mission is to develop safer and more effective RNA therapeutics, and that's not just limited to mRNAs, of course, but this is our current main focus in oncology, infectious disease, and autoimmunity. And we want to break down these technology barriers or help to break down these barriers that exist to address new therapeutic areas. And also, we want to make these more broadly available. And as we just heard from the earlier CEPI presentation, the stability and possibly decentralized manufacturing is quite important for infectious diseases. And I'll come back to that on the last slide. So we are obviously quite a small company at eTheRNA, but we have one advantage. We have been in this business for quite some time, so almost 10 years. So we have deep experience in mRNA chemistry and formulation. And we also, importantly, independent of some of the technologies we have developed by having our proprietary technology platforms and our own GMP manufacturing as well. So at eTheRNA, we're focusing on 3 technology pillars, not surprising, as pointed out by previous speakers, areas that are obviously looked at for optimization of these platforms. So formulation, the actual mRNA chemistry and process development. I have highlighted with the red boxes here the points I'm going to focus on in the remaining slides. So our proprietary ionizable lipids and how we tailor the LNPs for specific applications. And on the last slide, also the product stability and the decentralized manufacturing, as I mentioned. So I'm not claiming that LNPs are the only delivery platform or will indeed remain the only delivery platform going forward. But at the moment, it's certainly the most advanced and most established technology. And the COVID-19 vaccines have impressively demonstrated that, and also shown that the scale-up of to vast quantities is, of course, possible, although I'm sure this wasn't without hiccups either in some cases, but it is possible, of course, and that has been proven. As you probably all know, the composition of these LNPs are usually taken from 4 lipid classes to provide structural integrity and stability. And it's also known that there are various factors that can affect LNP properties. Obviously, the intrinsic chemistry of the lipids used, the ratios, but also the manufacturing process. So it's been recently shown in some nice publications that one of the intrinsic features of LNPs is the ability to immune stimulatory properties. So on their own, so without any encoding mRNA, you can actually, of course, activate or innate immune responses. And in some applications, like in cancer, this might be actually beneficial. But in other indications, like, for example, autoimmune indications, this is not desirable. So in our case, where we want to use LNPs in these applications, we are screening our libraries for minimal biological activation. We optimize these ratios, and we're also looking, in addition to that, into encoding factors that actively suppress the innate or the inflammasome activation. So if you project, the LNPs that we do on our platform and our current portfolio, you can see that we're developing several different LNPs, mostly for different routes of administration, but also within the same route of administration. So, for example, for the intramuscular route, we're looking at different biological activities which we're screening for to really tailor these LNPs for the applications in mind. Sorry, my slide is not moving. Okay. So how do we do that? So we deploy a systematic screening for our LNP optimization. We use our proprietary ionizable lipid backbone libraries, which we then co-formulate with the lipids that I just mentioned. We also have the opportunity to add functional components such as targeting, for example, which we're moving into, and we use a design of experiment approach where we optimize the design space for these libraries and LNP compositions, and then we screen those for the desired properties. So in this case, for example, we're looking at specific biodistribution we want to achieve, and you can see on the left-hand side with our IV LNP that we have data from non-human primates there that confirm that these LNPs are mainly going to the spleen, where we obviously targeting dendritic cells and activate the T cell responses. And on the right-hand side, you see an example of our intramuscular delivered LNP, where the LNP is mainly retained in the injected muscle, and there is minimal leakage to other organs in this case. So just to come back to the IV-delivered LNP1, so this has enabled us to progress our HPV positive cancer vaccine, which is going into the clinic anytime now. It's been approved, and you can see that this optimization has a strong effect on the CD8 antigen-specific responses on the left-hand graph, and this is directly translated then into increased efficacy in this syngeneic tumor mouse model here, which is HPV antigen positive. For our intratumoral injection, developing this platform has enabled us to develop alternatives to the sort of traditional cancer vaccine approaches by delivering mRNA that modifies and disrupts the tumor microenvironment. Essentially, you're looking, there's many options possible here. You can induce cell death, or you can create a favorable environment for activating T cell responses and attacking the tumor cells. In essence, it's about making cold tumors warm or hot. And you can see on the right-hand side an example of some of the mixes we have developed and the great responses we get in some of the tumor models used, in this case an MC38 standard tumor model here. And we have actually seen that in this application, we see a clear benefit of using LNP technology over, for example, naked mRNA injection in an intratumoral setting. And we have, within this model, we have also seen a long-term protection from re-challenge with tumors. And we obviously extending this to other tumor models right now. And this could be quite a promising platform. For our intramuscular LNP3 as an example here, we're showing some data from our SARS-CoV-2 vaccine. So these on the left-hand side, you see that we're generating good antibody responses against, in this case, delta spike protein and CD8 responses to the spike sequences. And this is achieved with a very good tolerability around the injection site. So we are progressing this as quickly as we can towards clinical study. So in terms of stability, we have been interested to improve stability for mRNA, especially with infectious diseases in mind. So we have used the LNP3, which is our intramuscular delivered LNP that is mainly used for our prophylactic vaccines, and lyophilized that, and we had some good successes here. We're achieving good lyophilized cake aspects, and on reconstitution, this is looking very promising. And on the right-hand side, you can see a whole body imaging study where we encoded obviously luciferase in this lyophilized vaccine, and you can see there's no difference really between the reconstituted and freshly prepared LNP formulations in this. So we think this has great potential for application in infectious diseases, especially in areas where obviously cold chain supply is a challenge. And on the last slide, I'll just briefly mention our activities in the manufacturing and process development areas. So we're looking to develop together with an engineering company called Quantum a small footprint continuous production system that can be moved around, that is offering you the option to decentralize manufacturing. And this will improve our capacity quite substantially in-house and offer obviously the opportunity to move this to areas where it's needed. And in addition to that, we are also, with our joint venture based in Nanjing in China, we are implementing the same capacity there. And from the middle of this year, we'll be able to produce at least these quantities as well from within China to supply some of our products and markets there. So this was quite a short presentation, but with that in mind, I would just like to thank you for listening. And we have a virtual booth at this conference. We are sponsoring that. So if you're interested, have a look there or get in contact with me to ask questions. And yeah, I thank you again.
Thanks, Volker. Very interesting, and putting a different focus now from the mRNA modality to delivery, which certainly is a key challenge for the further development. And so there's a question from the audience: where do you see the development of novel delivery technologies for the future? Especially, do you see anything besides LNPs? I mean, you're focusing on these now, but do you see further opportunities?
Well, with concrete, no, I haven't got a specific example yet. I mean, there's obviously people are working on that, and we're also, trying to engage into that. It's probably not a quick development, as you know. The development of LNPs has taken a long time. And some of the SARS-CoV-2 vaccines were only enabled because the technology was already available and could be licensed. But there is a lot of development behind these LNPs, and I think the same is true for alternatives. I think it will take some time to develop alternatives that are as safe and as efficient as the LNPs are. But I wouldn't say that LNPs will remain the only modality to effectively deliver mRNA or indeed other RNA modalities. I think there are, as I mentioned, there are some perhaps intrinsic properties of LNPs that are not beneficial in some circumstances.
Yeah. And maybe then as a last question, more technical, I mean, you showed the comparison between naked RNA and LNP encapsulated RNA for your intratumoral studies. And I mean, LNPs, they protect the RNA, they help with the delivery, they have immune stimulating activity on their own. So were you able to differentiate what was the main driver in your case to make LNPs the better way to do it here?
I think mainly the fact that you could probably deliver less mRNA, much lower doses, and as you say, protect the mRNA. And if you can if you can specifically retain the mRNA as well in the tumors, I think that's a benefit. We have the evidence that we have is when you use naked mRNA, you can use much higher quantities, and you still do not achieve the same efficacy that we've seen with the LNP delivery. We haven't fully explored that dose range, of course. Where the point is where you could achieve parity in terms of efficacy, but it's certainly lower in our hands. But there is obviously efforts going on outside eTheRNA that have shown that you can use naked or indeed other delivery vehicles for that in intratumoral settings. Yeah.
Yeah. And maybe as a last question before the break, it just came up. So what would be the scale of the quantum modular plant that you presented, and how portable is it, was asked here?
Sorry, the scale of the...
Of your quantum modular LNP plant, and how portable is it?
It's very portable. It's basically a couple of square meter footprint. It's a small unit that could be put in any sort of sterile suite quite easily. And obviously the scale would come from multiplying these systems. So you would have 2, 3, 4, 5 of these systems. And I think the advantage, as I understand it, is that these are continuously run. So it's not like a batch process that you run. It continuously churns out the product at a smaller scale, but because it is continuous, it's very efficient.
Okay, thanks, Volker. Very interesting. All the best to you and eTheRNA, of course.